Now, from the light microscopy, the next microscope is the dark field microscope. Dark field microscopy refers to the use of the microscope that excludes the unscattered beam from the image. What is the unscattered beam? As a result, the field around the specimen where there is no specimen becomes the dark, and generally the light which passes through the microscope and makes the objects very clear. Refer to the use of the microscope that excludes the unscattered beam from the image. Similarly, from here you can see the objective lens. First of all, the objective lens here, and then the stage where they put the specimen, and then the condenser lens. So in this way, we can see in the next slide, the dark field microscopy, it will become the contrast, and the background is very dark, so make the objective better and clear as compared to the simple microscope. Used for the live microorganisms that either are invisible in the ordinary light microscope, or maybe they are not unable to stain by the standard methods of the staining methods. So here you can see the purpose of the dark field microscopy. Here in the slides, it's very clear that dark field microscopy, the organisms are very clear, and in the dark background, maybe the most of the time we use the black background, and the images of the microorganisms become the light, and the light stares them, and they shows in this way. So its use, why it is used by because some of the organisms which we are unable to stain them with the standard ways of the staining, or we are unable to see them with the simple light microscope. Now, what is the principle of the dark field microscopy? Dark field microscope use a dark field condenser containing an opaque disc. The disc blocks the light that would enter the objective lens. Only the light that is reflected off, turned away from the specimen, enters the objective lens. And because there is no light, because there is no direct background light, the specimen appears the light against a black background, and then it becomes the dark field background, and that's why it is called the dark field microscopy. Here you can see in the figure, we can make it a little bit clear about the dark field microscopy. The light from here and the opaque disc on the way. Similarly, here is the eyepiece. You will put your eyes here, and you can see the light reflected by the specimen is captured by the objective lens. Here are the objective lens, which is the light will be captured by it, and it will give us the contrast by the dark field, and the images or objects or the specimens will be make it clear inside of the just like the light inside the microscope.